think it's about time we got started. So welcome to today's Oxford Centre for Personalised Medicine talk. Before I get started and introduce today's fantastic speakers, I just wanted to briefly introduce the CPM to anybody who's not familiar with us. So we're an outreach initiative based at the University of Oxford as a joint venture between the Wellcome Centre for Human Genetics and St Anne's College. And our aim is to communicate all aspects of personalised medicine with students, scientists, and to the public. So we host talks, events, we have a blog, and more recently we've been creating short video vlogs and also animations. And if you want to check out any of our previous talks, so including Jennifer Doudna, Heidi Ream, Adam Rutherford, and many others, then please head to our website. And you can also follow us on Twitter at CPM Oxford to find out about future events and also to tweet us about today's event. So today we are hugely fortunate to have two incredible speakers join us today. So Sonia Vallabh and Eric Minikel, and they jointly lead a research team at the Broad Institute of MIT and Harvard in Cambridge, Massachusetts in the US. And their research is focused on developing preventative drugs for prion disease. Now, I don't want to do a massive introduction and give too much away of their story. Instead, I'm going to pass it over to them to tell you more. So please take it away, guys.
Thank you. Thanks so much for having us. Huge honor to be here.
Absolutely. And as Nikki alluded to, we'll be talking about prion disease, how to treat it, how to prevent it. Hopefully we'll be able to do both. And before I get into the weeds of how, I'll just cue up how we got here and why we do what we do. So we're a married couple. We got married in 2009. These are some photos from our wedding day. And on the right, that's a photo of my mom helping me get ready that day. So she was 51 at the time, and she was healthy. And actually, she was organizing the whole party. She really did a lot and had a lot of energy that day and looked really happy. But what we couldn't have known at the time is that this was the last family event for which she would be with us. So about 6 months after these photos were taken, she went into a totally unexpected catastrophic neurological decline. And we didn't know what we were looking at. All we knew was that sort of day by day and week by week, she was losing her abilities, her ability to form coherent sentences, to get around the house independently and do simple activities. It was all just sort of falling away. We rushed around looking for a diagnosis. And despite seeing a lot of doctors at a lot of hospitals, we didn't get one in her lifetime. But when she passed away, and this was the end of 2010, we learned from an autopsy report that she had died of genetic prion disease. And this was a complete shock to us. And particularly hearing the word genetic, which was our sort of first indication that the story wasn't over. And that I was at 50-50 risk of having inherited the mutation that had caused my mom's disease. So this was quite a crossroads to come to as basically newlyweds. We had to make a decision whether or not to pursue predictive genetic testing. And we sought a lot of advice from people who we trusted, from family, from friends, also genetic counselors, physicians. I would say that it all kind of came down on the side of there's nothing you can do. This is an untreatable disease. There's nothing you can do to prevent it. How is this information possibly going to benefit you if you have an unlucky result? So consider just walking away. But I can't say that we really did consider walking away. I think we knew about ourselves that we couldn't go back to a time before we knew about my risk. So our options were to hang around in limbo, which was torture, or to just find out what we were up against. And so we chose to find out. And that's how we received this genetic test report at the end of 2011. So it's been a little over 10 years, and it wasn't the result that we hoped for. So we learned that I had inherited my mom's mutation, and sort of an outline of what that implied. So this mutation and a handful of others in the prion protein gene cause, with high reliability, fatal untreatable neurodegenerative disease. And these mutations, and we'll talk a little bit about penetrance, which we've since returned to as scientists, but what we were told as patients continues to be true for my mutation, that there's a near certainty of developing the disease in your adult lifetime. But an unusual thing about the tempo of prion disease is that it appears that carriers remain completely healthy until some unpredictable point when things turn. And from the very first moment of symptoms to dying of the disease is usually under 6 months. So this is kind of a characteristic thing of prion disease. I didn't realize when we were seeing it happen in my mom that this could be a characteristic feature of any disease for it to move so fast. But indeed, that is typical. So I couldn't have predicted what would happen with our test report one way or the other, but once we had it in our hands, things started to change. I had been trained as a lawyer and Eric had been trained as a transportation engineer before all of this happened to us, so we didn't have scientific backgrounds, but we found ourselves really highly motivated to learn more about what this meant. I think we were kind of startled to realize that after that very long clinical journey and ultimately like unsuccessful in her lifetime to try to get a diagnosis, that had sort of convinced us that this disease was so rare that nobody knew and nobody cared. But then to pull back the veil a little bit and realize that there were actually really active research groups around the world working on prion disease and working on prion disease therapeutics was kind of, it caught our interest. So we started off, sort of trying to educate ourselves and become savvy consumers of scientific information just to be informed patients or patients in waiting. And this took on a life of its own. So within a few months, we'd actually left our old jobs and we'd taken, sort of entry-level research jobs at Mass General Hospital. And from there, we started doing a lot of soul searching. Is this forever? And if yes, how do we pivot to focusing on our disease? Because if anybody's working on it, shouldn't it be us? We ended up starting a PhD program locally, and in that sort of journey to find a home lab, we're exceptionally fortunate to connect with people here at the Broad in Cambridge who took an interest in our mission and were willing to support us as trainees to really focus on prion disease and start moving in the direction that we knew we needed to go. So that's sort of what we've been doing ever since. And since 2019, we've sort of rolled forward our little research program here into an independent lab, and a lot has happened. So I think there's a long way to go in this quest, but I like to think we've also come a long way. So some of what we'll try to share today is the therapeutic strategy that we are literally betting our lives on. Drug development partnership that is many years in the making and hopefully soon to reach the clinic. Regulatory strategy has ended up being an interestingly large piece of this puzzle, as I think for many rare diseases it is. And finally, the people, the carriers that we're trying to connect with around the country and around the world to try to make prevention possible. Here's a picture of our lab. So this is something that we'll plug a few times. We're now a lab of 6. We're looking to grow. So if you folks know people who are looking for a postdoc position and might be inspired by this mission, if that's you, please get in touch. And here's sort of how we've organized what we'll present today. So basically, we've learned along the way that our mission is larger than developing or discovering a therapeutic molecule. There are actually a lot of different puzzle pieces that we need to assemble. And I think this is a mandate that falls especially hard on rare diseases, that you sort of need to take all of the different pieces that need to be put in place to have a successful clinical program, and you need to build it yourself. So we'll share proof of concept from preclinical studies in animals, but also what we've done to develop a biomarker, natural history, collect the patient population, and engage with FDA.
So I guess I'll start the first part. So just first off, what is prion disease? So you hear a lot of different names for prion disease, CJD, FFI, GSS. And there is some genuine diversity in terms of how prion disease can present in terms of the early symptoms, rate of progression. And there's also genuine diversity in terms of etiology, right? So it's the only disease that can be sporadic, genetic, or acquired. But in a way, all of this diversity and all of this sort of historical accident of different clinical terms for prion disease is just on the surface. At its core, prion disease is one thing. And we're on this campaign to convince neurologists to stop using terms like CJD, et cetera, and just call it prion disease in the singular, not prion diseases, but prion disease. Because at the molecular level, this is one thing. It's this protein that post-translationally misfolds, that misfolding spreads throughout the brain and kills neurons. And this is the pathway of disease. And that gives us a really clear drug target, right? So if we can target normal prion protein or anything upstream of it to lower the expression of normal prion protein, we think that that should be beneficial. And there's long-standing data to support this therapeutic hypothesis. So these data are, 25 years old at this point. I guess as background to say, the way we model prion disease in animals is by injecting prions into the brain of a normal wild-type animal. And this works because prion diseases are transmissible, and it's not just a disease of humans, right? This is a disease that naturally afflicts a lot of different mammals, deer and elk and cattle and sheep and goats and mink and camels. And so we can model it exactly the same in mice. So you inject prion disease brain homogenate into the brain of a totally normal wild-type mouse, and 5 months later, it develops terminal prion disease that looks spookily like the human disease. And if you do histology, the brain looks spookily the same as a human brain affected by prion disease would look. So they're not even models. This is the disease in an animal. So in wild-type mice, the disease takes about 5 months. But if you add additional transgenic copies of the prion protein gene, you can make the disease move faster. If you knock out one allele, it takes more than twice as long. And if you knock out both alleles, that mouse is invincible. It can never be infected with prions, never replicate prions, never develop symptoms. So the less PRP, the better. And luckily, we're not too worried about knockout phenotypes. So the knockout phenotype, people have spent decades looking for it, and it is somewhere between mild and non-existent. So there is a mild peripheral neuropathy in homozygous knockouts, but it's not seen in central nervous system, and it's not seen in heterozygous knockouts. So there should be quite a therapeutic window where you can lower prion protein enough to be beneficial for disease before you remotely begin to worry about this. And in spite of that, we always get this question, well, until you've seen homozygous knockout humans, how do you know for sure? Isn't there a reason we have this gene? And I always like to remind people, if that was our attitude, we wouldn't have any drugs, right? If your attitude was just because a gene has a function, therefore we can't develop an inhibitor or a suppressor, well, then we wouldn't have statins or aspirin. There's actually lots of really successful, safe drug targets that are even essential genes. And prion protein isn't even close to essential. It's as non-essential as like an olfactory receptor or something. It's among the most tolerant of loss of function variation of any gene in the genome. So we're in this really lucky position. This gene is essential for disease and dispensable for life. So our question is, how do we lower it as much as we possibly can?
So I'll share one strategy, and this is perhaps the furthest along and most likely to reach the clinic in the near term. Of the different ways we can think about doing this. So to back up one step, details of the therapeutic strategy, as Eric told you, we want to reduce prion protein. We want to do this across the central nervous system to the greatest degree that we can. We have a non-allele specific strategy. So although I carry a mutation in the prion protein gene, we're not targeting the mutation per se. We really want to just tamp down all the protein. We need to do it across regions. We don't have a region that sort of converts first. And we need to do it primarily in neurons, although we can talk about the advantages of potentially targeting broadly across cell types. So antisense oligonucleotides, I'm sure that you folks have some familiarity with this modality. Just to introduce at a very high level, so these are short strings of chemically modified DNA that can sequence specifically bind target RNA. And one of their possible mechanisms of action is to trigger the degradation of that target RNA. So can we reduce levels of prion protein RNA, thereby protein, We first came to this strategy in 2014 was our first conversation with Ionis, and some data were just starting to emerge around being able to deliver ASOs across the brain. This is a big challenge for us. It's just achieving the distribution that we need and being able to do that safely. So we launched this collaboration, and here are some preclinical ASOs that target the mouse prion protein RNA and are lowering its levels by about 50%. And here's the mouse model that Eric described to you. So we infect the mice with prions. They go on to really, truly develop the disease. So with all that preface, I'll show you a bunch of survival curves, but I'll actually move through them pretty quickly because they all tell, thankfully, a pretty similar story. So this was our first mouse study that we ever did. And the treated animals are in green and blue. They got 2 doses of ASO 90 days apart. And they outlived the control animals by about 60 to 80%. Really clear result. And our sister lab at NIH sort of did at the same time a study with very similar design and also saw a really similar survival benefit. So we were able to ask the question with these ASOs, how much do you need to lower PRP to start to see a benefit? And in this dose response experiment, the lowest dose lowered the prion protein RNA by 21%, and it gave us a 15% survival benefit. And the more we lowered, the better survival benefit we got. So this does look like sort of a continuous dose response where, you know, any amount that you can lower PRP is beneficial, but the more the better. And in this experiment where the animals were dosed early, we could see that it wasn't just a dose responsive extension of survival that they enjoyed, but it was actually a dose responsive extension of healthy life. So they maintained body weight, they took longer to accumulate symptoms, they built normal nests for a longer time, sort of across all these different measures. So that's sort of the early treatment picture. And then we moved to treating animals at a point where prions had had a chance to establish a hold on the brain and to establish some neuropathology. So here animals, we waited until they had sort of accumulated some signs of neuropathology and then gave a single dose of ASO. And on the left, the single dose was able to drive down these rising levels of neurofilament light chain, a marker of neuronal damage in plasma. On the right, these transgenic animals have a sort of bioluminescent readout of reactive astrogliosis, which is an early pathological sign in a prion brain. And as that signal went up, we gave a single dose that brought the signal back down. So some indication that even a single dose of drug can reverse neuropathology. And here we did a sort of a big study looking at chronic dosing. This is the most aggressive treatment paradigm we've ever tried. And animals started to get chronic dosing every 90 days, either early, before the emergence of neuropathology, or late, which here means sort of at the cusp of symptom onset. So you can see that both paradigms do a benefit, but the difference is pretty striking. These late animals have sort of a modest survival benefit, but early animals, we tripled their survival. So we basically did for them what deleting one copy of the prion protein gene can do. Here we go even later. So these are animals that had already started losing weight. They're measurably sick, they're symptomatic. And when we give a single dose at this time point, we can do a significant benefit to a subset of the animals. But the nature of that benefit is different. Earlier, we talked about extending healthy life. Here, what we're extending is the symptomatic phase of disease. They don't gain weight back. They don't lose the symptoms that they accumulated. And if we push even later to a point where some animals have already reached terminal endpoint, there is such a thing as too little too late. We can't benefit any of the animals. So at a high level, what have we learned? I think we've convinced ourselves that prion protein lowering really works in this disease in a variety of paradigms. But when you treat really matters. And I think that has implications for what we do in the clinic.
So one of those implications is it really drives home the importance of a biomarker in order to have something to measure in pre-symptomatic individuals. But I think Sonia's later on going to talk about sort of the clinical and regulatory pathway and how exactly we frame that up. We were thinking about a biomarker for this therapeutic program from pretty early on, actually from our very first meeting with Ionis Pharmaceuticals. Holly Kortashevich there, who really became a fantastic scientific mentor to us, said, we as a company would never go after a target where there isn't a way to measure target engagement, right? Where we can't see that our ASO has even done the job that it's designed to do. And so you guys need to come to us with a pharmacodynamic biomarker. So again, pharmacodynamic biomarker is just measuring a drug's intended molecular effect. That's distinct from a disease biomarker, which is measuring, the pathology or the disease state. And for those who don't think about this every day, I should back up and say, biomarker is something you can't feel, right? So think of things like LDL, viral load, blood pressure, et cetera, distinct from a clinical outcome, something that affects the way a patient feels or functions. We started from early on thinking about, could measuring prion protein in cerebrospinal fluid, or CSF, be a pharmacodynamic biomarker for this therapeutic hypothesis and this drug modality? Now, in some ways, we were really lucky, right? You could easily come to a target where no one's ever measured it. There aren't even good antibodies for it, et cetera. And here we found that there was a commercial ELISA kit that you could just order from VWR. It's off the shelf. There had been a few papers already published using it. We knew that PRP was abundant in spinal fluid, tens to hundreds of nanograms per mil. But we had some serious challenges in how to work this up as a biomarker. One is that there was evidence that CSF PRP actually goes down in symptomatic prion disease. Paradoxical, because this is a gain of function disease, and we know that total prion protein is accumulating in the brain during the disease. But this is also true with amyloid beta and Alzheimer's, right? You get plaques in the brain, but actually less of it is soluble and shed into CSF. So we thought it could be something analogous like that. The question is, does that confound how you use this to measure the effect of a drug that you hope is going to lower the protein, but thereby alleviate the disease process that caused that lowered amount in CSF in the first place? The other challenge is that in these published papers using this assay, there was enormous variability in CSF PRP concentration from person to person. And by enormous, I mean like 100-fold, right? One person would have 2 nanograms per mil of prion protein in CSF, and another would have 200. So why is that, and is that going to be a challenge? So we started running just kit after kit of this assay. And one thing we noticed quickly is that you have the same sample aliquoted to different size in Eppendorf tubes. You get a different readout of PRP concentration. And that made us think maybe this protein is really sensitive to what we call pre-analytical artifacts. So what happened to that sample before it was analyzed? And so Sonia did this battery of experiments looking at neat CSF versus if you add a little bit of detergent to try to keep the prion protein in solution. And lo and behold, the more that you pipette or move it between different tubes, basically every time the cerebrospinal fluid touches plastic, you lose half the prion protein that was in it. It's just a really sticky protein that loves to stick to polypropylene. And adding a little bit of detergent almost completely mitigates the problem and keeps it in solution. It's not enough to just use a different material. People always ask, well, what if you just use glass for everything? Actually, glass was even worse. So it's a really sticky protein. It's a finicky analyte, and we had to come to terms with that and develop protocols that would allow us to handle it. And so we proposed that going forward, when people are doing spinal taps specifically to collect spinal fluid for prion protein quantification, we basically said, you should be this fussy about how you handle that CSF. You add detergent really early on, and you always aliquot the same volume into the same tube. You always pipette every sample up and down the same number of times. People said, you guys are crazy. If it takes this level of finickiness to get your analyte to work, then you don't have an assay. And we said, but this is really important. We need to be able to measure this for this to work. And we have gotten that protocol to work. It's been implemented clinically. The other thing that we had to look at is, do we think that PRP in spinal fluid actually reflects what's going on in the brain? And luckily we found there's tons and tons of PRP in the brain. There's next to none in blood. Those are the two main sources of proteins in spinal fluid generally. And sure enough, spinal fluid prion protein was in between. prion protein is not correlated with hemoglobin. You spike blood and it doesn't make a difference. So we're confident that prion protein we measure in spinal fluid is coming from the brain and should reflect the brain. And most importantly, when we looked at samples that had been collected uniformly and carefully in the same person over time, we didn't see that 100-fold variability, right? So you look at, these are samples from the same individual at week 0 and week 8, spot on, same amount of prion protein 8 weeks later. So even though there had historically been this huge variability in samples from one person or another, from one center or another center, if you handled samples carefully, a person had a steady baseline over time. And that's reflected here where the coefficient of variation for, historical cohorts where they came in different sized aliquoted tubes and so on was enormous, but carefully handled samples, we could get it down to 13%. Our study also confirmed that CSF PRP does go down in symptomatic disease. And we wanted to know, is there any chance that that's just an artifact, that the protein is misfolded, cleaved, the epitopes aren't accessible, and so ELISA isn't picking it up? So we actually developed a whole new assay. We developed this targeted mass spec assay. We said we're going to look at tryptic peptides from 6 different positions, spanning all the way from the N-terminus to the C-terminus, and quantify those by MRM to get a totally orthogonal look at whether this protein is really going down in spinal fluid when people have symptomatic disease. And for all that, the answer actually ends up being exactly the same. So for every peptide we look at, prion protein in spinal fluid goes down when people develop prion disease. The nice thing about this, so that didn't solve our sort of disease state confounder problem. But the nice thing is, wow, PRP is a really simple analyte. Anybody who's studied tau knows you have some of these proteins where there's different isoforms and they behave differently in spinal fluid. It looks like we don't need to worry about any of that. This is one analyte, and any which way you measure it, you get the same answer. So to skip over a lot of additional work, we went back and developed a whole new assay that we deeply validated, like to FDA standards. It's cross-species reactive. And we started using that ELISA assay for preclinical studies. And we recently published this work validating that in rats, where we dose with an ASO, and then we measure total prion protein in a whole brain hemisphere or in spinal fluid, we validate that we knock down prion protein in the whole brain, and that is read out in spinal fluid. So we have a sampling compartment that's responsive to the drug. So we've been amazed. It's been a real journey for us to realize, from thinking, oh, there's a way to measure this protein, problem solved, right? Actually, it's been years and years of work to get it to where we can say, this is a pharmacodynamic biomarker. If you knock it down with a drug, we should be able to read that out. So I think the main question that would be left open by everything I just told you is, do we need to worry about disease-related decline in CSF PRP? Is that going to make it difficult to use this biomarker in a symptomatic patient population, but could we use it in pre-symptomatic patients?
And to answer this question, we launched a natural history study. So this has now been ongoing since 2017 in collaboration with Stephen Arnold at MGH and his great team. And we've basically been recruiting PRNP mutation carriers who are currently healthy and non-carrier controls. And the study is ongoing. I hope we'll keep learning from it, but I'll share some of the highlights of what we've learned so far. So if you've studied a different neurodegenerative disease that is more slowly progressive, these data might be surprising, but we looked at CSF total tau and CSF neurofilament light, so classic markers of neuronal damage in our carriers and controls. You can see in red that in symptomatic prion disease, these markers are off the charts. Neurons are just dying at an incredible rate. But our carriers and controls are indistinguishable from each other, and they're all in the normal range. And we don't see any sort of, suggestive trends over time that are carrier specific. So it does look like maybe neuronal damage is not happening sort of prodromally at a detectable level in the average carrier. We see the same thing when we look at neurofilament light in plasma. Everybody appears to be normal. We've also had the chance to look at a prion specific assay. So this is sort of a prion detection assay called RT-Quick that's used diagnostically. And basically it can pick up prion quote unquote seeds and read them out in a fluorescence curve. And that's what you see in panel A. These are again positive controls where there are a lot of prion seeds in the brain and in CSF that are picked up. When we look at our carriers, by and large, they are negative. suggesting that prions have not yet started to form in their CSF. So maybe that like very inception of the disease process has not yet occurred. We have seen one individual who was healthy but RT-QuIC positive. Their total tau and NFL were normal at that time. And they remained asymptomatic a year later. So this is something that we're still following up on to understand the prognostic value of this information. So where does that put us with pathological biomarkers? I think we're probably not going to plan a trial around them in these sort of pre-symptomatic carriers because too few people actually have signs of pathology. There are other sort of technical reasons why it would be hard, but I think when I look back at our mouse data, maybe the best reason to not use a pathological biomarker to sort of recruit people to a trial is that that's not when we can do the most good. In our mice, it was when we treated before pathology that we tripled survival and had like a really profound impact on their outcomes. So I'll shift now to talking, sort of picking up the thread of prion protein levels in CSF. So this is something we've also had the chance to look at in our natural history study. Eric showed you data from historical samples that weren't from sort of, prion protein mutation carriers. Here, we sort of replicate those findings in our cohort. So when people give CSF samples 2 to 4 months apart, whether you're a carrier or a control, you have this nice stable baseline of prion protein levels. So we think that a drug-dependent decrease should be easy to measure. And that trend is maintained even out a year, year and a half. So I think this is actually really lucky. You can see in gray those historical controls that Eric alluded to, where the samples weren't handled with extreme caution and care. So that continues to be really important, I think, to our program going forward. So CSF PRP behaving well, carriers, they appear to be just healthy. And I think that some people might frame as a negative result. We think it's an amazing opportunity to hopefully treat people to keep them healthy.
So how do we go, what is this patient population and how do we do trials in this population? So we think of prion disease as having 2 patient groups. There's the symptomatic patients, not diagnosed until they're sick. This would be all sporadic patients and some genetic patients who hadn't had predictive testing, didn't know they were at risk, et cetera. And then pre-symptomatic, so people like Sonia who might have a genetic test report decades before onset of disease. So in the symptomatic patient population, there is precedent for doing randomized trials and following people to a survival endpoint. So I think that shows that this is feasible. I think these examples also show some of the challenges in that approach. So in particular, this doxycycline trial in France and Italy, half of the patients were already either intubated or ventilated at the time that they were randomized. So to me, that raises tough questions about what does it even mean to do good for a patient population that's already that impaired? And I think reasonable people have different answers to whether they would want that for themselves personally. But I do think we should also grapple with, would a molecularly targeted therapeutic still be relevant at that disease stage? Or is that a stage where secondary pathologies are now driving people to progression? So I think there's assets, we have a good molecular diagnostic, RT-QuIC, and there's good referral networks in the US and in the UK and many other countries. But we need to catch people earlier. And so we don't want to put all of our eggs in this basket of hoping that a symptomatic trial will work and deliver us a drug that would help for people like Sonia. So we need to think about the pre-symptomatic population. So Sonia alluded earlier, we did this study, 5 or 6 years ago now, to try to characterize the genetic population and figured out that a lot of mutations that people thought were pathogenic are actually low penetrance. But we identified a subset of mutations that really do seem to be, 90+% lifetime risk, unfortunately, including Sonia's mutation. Even just the top 3 of those variants account for about 85% of cases. So we've defined a patient population in which there's high enough risk that we would argue it's justifiable to go in with an experimental therapeutic. That risk-benefit balance is there. But the question is, how do you do a trial in those people? What do you measure? So one precedent is in genetic Alzheimer's, there's folks doing this trial where you take totally healthy mutation carriers with no requirement of pathology. Your only sort of ticket into the trial is your genetic test report. And then you get randomized for 5 years to a cognitive endpoint. And because that precedent is out there, we wanted to model whether that would be possible in prion disease. In other words, take people like Sonia, randomize them to onset. Now, I would have a lot of feelings about what's basically, randomizing people to the death. But the question here was just, is this even numerically feasible? So we went out and gathered data from 9 different centers with over 1,000 individuals with age of onset, death, or last follow-up, and basically built life tables saying, what is the distribution of when people have onset of this disease? And it turns out that the age of onset is super variable in prion disease and nothing predicts it. So for Sonia's mutation, there's been a case as young as 12, and there's been someone who lived to 89 without developing symptoms. So it's an incredible variation. We don't have any way to predict for any one person when your onset will be. And that actually is a huge problem for this idea of randomizing people to a clinical endpoint. So one way of thinking about it is that, so here the band thickness is how many patients there are, and the y-axis is what is the probability of having onset in any given year of life as you age. And basically, if you go early, there's lots of patients, but their annual risk is low. And if you go late, annual risk is high, but there's not many people who live that long in the first place. So basically, there's no place you can draw a line and say, give me all the patients older than X years of age, and I'll have a cohort where I expect enough of them to develop disease in the next few years that I can power a trial. And we modeled that out with a power calculation, basically saying, under reasonable assumptions, you would need like 800 people to be randomized in order to power a trial, and that is more patients than we have in this rare disease. So that's just a non-starter. That's not a trial design that we're going to pursue. And the fact that that is numerically impossible actually has policy implications, right? So it's in US law that FDA shall consider instances where the low prevalence of disease renders, more traditional clinical trials impractical. Then this accelerated approval pathway should be considered. So basically, is there a way that we could give provisional approval based on a biomarker endpoint and then do a post-marketing study later on to confirm clinical benefit? So I think that's where I'll hand it over to Sonia to talk about that path.
Absolutely. So background on accelerated approval, instead of sort of, randomizing to a clinical endpoint, which is, a change in how a patient feels or functions or survives, we would be trying to use as our endpoint in a trial, a biomarker that is reasonably likely to predict clinical benefit. So a classic historical example here is HIV viral load in AIDS. And as Eric alluded to, there are sort of requirements for when you qualify for this program. It doesn't free you from having to show efficacy and safety. And you do have the obligation to do a phase 4 trial in which you ultimately, in sort of a post-marketing study, confirm clinical benefit. But when we look at the list of when Congress thinks that this pathway should apply, I do think that we check the boxes, including that this is an unmet medical need, that rarity makes traditional trials difficult or impossible, and that the risk benefit is favorable. So, I talked earlier about the opportunity that I think we have to keep people healthy through a primary prevention strategy. And our proposal for how to do that is that prion protein levels in CSF should be that primary endpoint. If we can lower prion protein levels in the brain, then we should lower them in CSF as well. Eric showed you the animal data supportive of that idea. And this is right on disease pathway. We talked earlier about all of the proofs of concept, genetic and pharmacological, that if we can lower PRP, that should be predictive of a better outcome. So we took this plan to FDA for the first time in 2017 and went in super terrified, and we're actually amazed by the reception that we got. I think we expected to really have to convince the scientists in the room, and instead what we heard from them was, of course you want to prevent prion disease because it's basically a lightning strike. You can't wait until you're struck. If you have genetic information ahead of time, we need to have a way to act on that. And it makes sense that prion protein could itself be your biomarker because it's the pathogen. It is just the core of the disease. So instead of, hashing out whether this is a good idea, we need to talk about how to get it done. And they have sort of continued to advise us since then. And I will say, it's a lot of boxes to check. There is a lot that you have to do to sort of validate a biomarker and a therapeutic strategy to the level that will support this kind of unconventional trial design. But we're working hard on getting that done.
So I guess I want to end on this note of primary prevention. I think it is, as patients, to us, it's obvious, right? We of course, from the moment we set out on this journey, the goal was to keep Sonia perfectly healthy, right? It's not that we wanted to extend that state of dementia that we saw in Sonia's mother. So you could ask, if this is what patients want, why isn't it the norm of how we do drug development? Why aren't more trials primary prevention trials? And we've come to appreciate there are some genuinely difficult questions that primary prevention raises, right? You need to be confident that you have patients that are high enough risk that an experimental drug is justified. We're constantly having to answer the question, let's say this works, well, when do you begin treatment? Now, our answer is, it should be just based on a genetic test report at some age where your risk is high enough based on those life tables. But I think the pressure to wait until it's too late, to wait until you have a symptom or wait until you already have neuropathology we can detect through a disease state biomarker, that pressure is always going to be there. There's always going to be people saying, can't we wait a little while before we reimburse this? Can't we wait a little while before we take this risk? It also constrains your universe of potential mechanisms of action. So I think we're really lucky in prion disease that, lowering PRP is just the therapeutic strategy that the genetics and everything points us to, and that strategy is relevant in a person in whom the disease process hasn't started. But you could imagine, there exists therapeutic hypotheses for many diseases where treating before the disease process has started, you're not doing anything and there's nothing you can measure. And then finally, what do you measure in a trial? What's the outcome, if not a clinical benefit? How often do you have that alignment of there's a biomarker that's measurable, relevant to the disease process, you can validate the assay, the relevance is unimpeachable, and so on. So it is really a tough package to put together. And I think we've been blessed that as much as prion disease has a lot of constraints that make us not want to put all our eggs in the basket of a more conventional randomized trial in symptomatic patients to a clinical endpoint, prion disease is also an opportunity, right? This gives us a chance to pilot this primary prevention model in a case where we would argue all of those stars align, right? We actually have an opportunity to take perfectly healthy people, keep them healthy with a genetically targeted therapy and a biomarker readout. So even though prion disease is a pretty unique disease, hopefully if we're successful, there's some inspiration and some lessons here that could be applicable to other diseases as well. I guess one final reflection, so Sonia sort of opened by saying, here's the 5 puzzle pieces to make this disease developable. And when we started becoming scientists 10 years ago, we thought the whole battle was find a drug and that's all that matters. And it was a real evolution for us to realize, actually, there's companies out there with platform technologies, ASOs are just one example, where they could apply it to targets X, Y, and Z, and figuring out how does your target map onto one of those platforms and how do you convince them to work on it actually can be a much quicker route to getting a drug than trying to discover something yourself. But you have to have all those puzzle pieces in place to make the disease what industry folks would call, quotes, developable. And so, without taking more time to get into it here, there's a couple of blog posts here that sort of dive into why are those the puzzle pieces that are necessary, and why is that what the folks who may hold in hand the technology to cure your disease, what they're looking for, and why is that a quicker path than trying to find that molecule yourself. So thank you so much. Again, really honored to be here, and we would love to take questions.
Perfect. Thanks so much, Eric and Sonia. That was fantastic, as always, but it's been a pleasure to kind of follow some of your progress with all of this as you've been going along. Just for anybody listening, just wanted to, Eric just pinpointed us a couple of his blog posts there, whereas actually I'd recommend you go and check a few more of them out, actually, because Eric has a fantastically clear way of explaining a lot of concepts. So if you've not heard of ASOs before, head to Eric's blog, and there's some really great explainer posts over there. So That's really great. So we've had quite a few questions come in, so keep them coming. I'm kind of going to roughly split them into ones talking about the kind of science and more the kind of personal ones. So I think we'll start with some of the science ones. So firstly, you spoke about some of your work on penetrance, Eric. So somebody's asked, do you think the incidence of high-risk prion variants could be underappreciated?
I think that it is underappreciated in the sense that, you ask how many people in the US hold the genetic test report like Sonia's, probably 300 or so. But how many people have a high risk mutation? We think that the prevalence is probably closer to 1 in 50,000. So there should be thousands of those people in the United States. So I don't think that the prevalence is going to turn out to be wildly higher than that 1 in 50,000 that we're now estimating, but I think that that figure itself is not appreciated by most people and not always reflected in the number of patients that we can reach today, right? So I think that gives us a target for how big could the market for a drug be eventually if we can get people to come out of the woodwork. But there's sort of this whole funnel of filters people have to pass through to get where Sonia is sitting today, right? So first, you're not going to just go looking for this apropos nothing. It's only going to happen because you lost a loved one to the disease. So that doesn't happen for everyone. If you're de novo or incomplete penetrance, somebody died young of something else, then they have to be diagnosed correctly, which I think we're doing better at today, but still underdiagnosed, especially here. I think you guys do better in the UK. And then especially historically, right, you ask somebody whose parents died 20 years ago, much less likely that they were diagnosed correctly. And then do you pursue autopsy, find out that it's genetic, find out that you're at risk, and then conditioned on finding out you're at risk, do you want to know? So through all these, I think we're getting well under 10% of the people that are out there. And hopefully that's enough for us to do trials, but we have a lot to aspire to in terms of once we have a drug, we have to use that to convince people this is actionable. You should go out and find out about this. You should diagnose this more accurately and more quickly. There's a lot of work to do.
Sorry, I'm talking and whilst muted, talking to myself. So a couple of questions based on your ASO strategy. So somebody's asked, how deep do these ASOs penetrate within the brain?
Yep. It's a great question. I think distribution is always a big challenge and a big topic for us. So the strategy is to deliver via CSF. So in our animals, we do intracerebroventricular delivery through the skull. In humans, it would be intrathecal at the base of the spine. There are data now from non-human primates. There's some limited data from human tissue too. And basically, it looks like you have a gradient, as you might expect, based on the regions that are better served versus less served by the CSF flow. So we think we can get really reasonable knockdown in the cortex, maybe along the lines of what we saw in our animal studies, but that as you get into the deeper brain regions, and particularly sort of like thalamus and striatum and cerebellum, probably it's going to be more limited. We don't think it's zero. And so from what we've seen from our mouse studies, where even a 20% knockdown gave us significant benefit, I think if we can achieve 20% knockdown in deeper brain structures, that will still be beneficial. But this is definitely an area where, whether it's sort of new and improved ASO delivery or ASO chemistry or other technologies altogether, I think it's an area for improvement.
Yeah, great. And another one on kind of ASOs. So you've mentioned that PRP is not seen as an essential protein. However, do you have an idea of the side effects that you could observe in a trial following use of an ASO against PRP?
Yes. So I think based on all the knockout models, the thing that will be monitored closely is like any sign of peripheral neuropathy, which has been seen in knockout mice. And the sort of histological correlate has also been seen in knockout goats, although they didn't have a phenotype. This phenotype is pretty mild, it's age dependent, and you kind of have to draw it out by doing the right test, so it's not overtly behaviorally obvious. So we don't think, even if we were to see it, that it would be a significant concern. And in particular, it's only seen in homozygous knockout mice, not heterozygotes. I think heterozygotes are a better model, for better and for worse. They're a better model of what we can achieve with today's technology. That's really what we sort of where our minds go in terms of on-target side effects, then I think you have to wonder about sort of off-target and platform side effects and liabilities, which will be sort of more generic across, for example, all neurological ASOs.
Yeah, great. So you've spoken about the kind of ASO strategy. Somebody's asked what are your thoughts on employing gene editing, so CRISPR, to inactivate the gene at the DNA level, and what are the main challenges around that kind of approach?
You give us a way to deliver it,
Yeah.
we're there.
I mean, we love it. We just got to get it there.
Yeah, it's all about drug delivery.
Yeah, great. And somebody else has asked, could analogous strategies be used to Parkinson's or motor neurone diseases?
Could be and are. I mean, you know, actually they're ahead of us, right? There's ASOs in the clinic right now against synuclein and LRRK2.
Nice. Okay. So on a slightly different tack. So if prion disease doesn't look like a Mendelian monogenic disease, are there other genes involved and identified through GWAS that could be targeted therapeutically?
Yeah, so thanks to Simon Mead's group at University College London, there's now 2 GWAS hits other than PRNP. So they're STX6 and Gal3ST1. I think we don't yet know how therapeutically relevant those could be. I think, you know, we've known about PRNP as the target in this disease for, you know, 30 or 40 years, and we're only now beginning to make good on that as a target. So I think we're probably a long ways from taking advantage of these other new targets. I think they're interesting, but you know, things like direction of effect, at what disease stage are they relevant, et cetera, still need to be worked out.
Great. Another question on kind of genetic basis here. So if it is genetic, is there expected to be any genetic anticipation? So earlier onset, as it's inherited from generation to generation?
Luckily, no. And there actually are reports out there claiming that there's anticipation in genetic prion disease, which I think was very alarming for our community. And then Eric did a study in 2014 where he showed that that was sort of ascertainment bias artifact. So thankfully, I mean, when we say that age of onset isn't predicted by anything, that includes that we don't see anticipation.
Great. And somebody else has asked, is there anything known about the initial triggers for the misfolding of the prion proteins?
Yeah, this is an absolutely classic question that our field is asking itself all the time. And the unsatisfying answer is that we don't know of any trigger. And I'm not convinced that we'll ever be powered to discover one. But I also have a sort of like a personal pet theory that it could simply be stochastic. Because we know that anyone in the general population, we all express prion protein. And in any person, even if you don't have a pathogenic variant, it could happen to go wrong at some point in your life. It's just this floppy protein of which you have so many copies. And it could just happen to, you know, unluckily flip into a self-propagating conformation. If you have one of these pathogenic variants, that's much more likely to occur at some point in your life. But I think there could still be a randomness around when. So there may be no answer.
Great. So I'm going to change tack and go on to some more kind of personal or career related questions. And it's a good moment to say that there's a lot of people saying that your talk was very inspirational in the chat. So first, can you please talk more about your process of deciding to start your lab at your institute directly after your PhD, and how you've digested such a huge amount of information across different sectors in just 12 years?
I guess the decision to stay at Broad, I mean, we had already just been in this like incredibly unique and fortunate position to sort of start a new research program as PhD students. We were embedded in Stuart Schreiber's lab, and he, you know, has a long, illustrious history in chemical biology, but had never worked in the sort of neuro space, but was interested. And so we sort of like became this little like hub within his lab that was looking at neurodegenerative disease, and having sort of been able to launch that, including, you know, that as PhD students, we actually set up our own research space here, you know, where we could work with prions at the appropriate biosafety levels, and we had all the right protocols when we started a prion animal colony, which Broad had never had before. So there was just a lot of infrastructure that we were able to build at a really early career stage, which was so lucky. And then sort of having gotten it all off the ground and having found such just like unbelievable institutional support for this work here, sort of felt like, maybe for many people, sort of switching institutions and taking a little bit of time to start up and rebuild in a new place would be a totally acceptable sort of path that could have a lot of advantages. I think for us, we're looking at the clock thinking, we hope to be in the clinic this year. It sort of feels like every month, of productivity really counts. And so we wanted to roll forward what we'd built.
Nice, that leads me very nicely into the next question. So has your extremely personal link to prion disease increased the speed of your effectiveness in your investigation? And have there been times when the link was actually counterproductive, for example, biasing the way that you've interpreted your results?
We got this question a lot early on, like, can you be objective prion scientists with a sort of personal mission? And I think that people were concerned that we would be overly optimistic. And I have found exactly the opposite, that in fact, everybody has some inherent bias to to hope that their experiments are going to work, but we are the very first people to throw something in the garbage if it's not working, because, the fact that lives are at stake, including mine, is just never far from our minds. I think if there's sort of a related way in which the mission has made us more effective, it's that we are really aggressive about pruning the tree. And, this is a sort of pros and cons way to be in science. I think sometimes if you're able to follow an interesting chance finding, your career can take you in really unexpected directions, which can be beautiful. But here we are, like, not open to going in any unexpected directions, solely on mission. And so we do keep a really lean portfolio of projects. And I think that that's helped us drive them forward fast.
Great. So somebody else is asking, it's very intriguing that you went from non-science jobs to doing scientific research. So how did you move from these jobs to science, and what was your experience with that process?
We definitely started at the bottom of the totem pole. Yeah, I mean, your first job was stem cell technician.
Yep.
I came in sort of an entry-level bioinformatics role. It was hard. I mean, there's a lot to learn. It's both that we were changing careers and this was an area we didn't know. It's also that science, there is a lot more to know than there was in our old careers.
Yeah, I think so.
And sure enough, here we are 12 years later, and it just is this like lifelong learning process, right? I think, I don't know, my experience in science, you never stop feeling like you're behind and there's so much more that you need to know that you don't know.
That's very true. Next is, what would you say is the biggest logistical hurdle that you feel you faced whilst attempting to translate your research into an actionable outcome?
I mean, we're still not in the clinic.
Yeah.
Stuff is slow. Drug development is slow, right? It takes a lot of time to really make a drug, right? There's a lot of proof of concept and a lot of drug candidate selection and manufacturing and good laboratory practices, toxicology and regulatory filings. It just, to really make a drug is a long, long process. And, thank heavens we got that genetic test report when we did, but there's still a lot of jeopardy. Will it all be soon enough? And I think we're constantly looking at the calendar being like, how could this move just a little bit faster? Is there anything we can do?
I guess that's a nice segue into the next one is, do you sometimes wish you could just switch off and work on something else?
Sure. I mean, I can't say that it's never crossed my mind. And we've been asked, actually, oh my goodness, by people who, like, bless them for their optimism. What will you do after you succeed and you have a drug for prion disease? Will you just go off and do something completely different? Which I think that the answer is no. Even like entertaining that hypothetical, the answer is definitely no. I mean, I think we are invested now in the way in which we're able, hopefully, to serve this disease community. And having learned more about sort of the drug discovery process, we are haunted by the limitations that even a very effective first drug will have. And I think the mandate to do better, whether it's doing better at discovering an effective drug or delivering that drug or diagnosing the people who need it or like reaching the pre-symptomatic community. I think there are going to be so many directions in which we can continue to improve just forever.
Perfect. And the final question is just a logistical one, which will the recording be available afterwards? Yes, it definitely will. It'll be shared on our Twitter. Apparently we're aggressive on Twitter, according to Eric and Sonia. It will be aggressively shared on Twitter. So please circulate it to anybody else who you think will be interested. And just remains for me to say, thank you, Eric and Sonia. That was truly inspirational, as always. You guys are amazing. Keep doing what you're doing. I have confidence you will succeed. And hopefully I will get to Boston and see you both soon.
Well, thank you
Thanks
so
so
much,
Nikki. Really appreciate the
Thank you.
invitation. Great talking to all of you.
Thank you.
Bye.